the strength from many subjects like the basic subjects like microbiology, biochemistry, molecular biology, and then also it mixes up the concepts of advanced subjects like genetic engineering, gene editing, and also engineering principles in general. And today we are also trying to harness the power of artificial intelligence and synthetic biology, and then we try to apply all the knowledge that we gain out of this interdisciplinary, multidisciplinary approach into various industrial applications and also in the field of agriculture, medicine, and environment. So now another query was about what is the difference between monotrichous and lophotrichous flagellation. So as the keyword here is the flagellation, so the presence of flagella. So not all microbes will be having flagella, but again those who have flagella, those which species which have flagella, some of them may have one flagella only. So that's what the word mono stands for. So monotrichous bacteria contain a single flagella at one end of the cell. So and you have the lophotrichous which contain two or more flagella. So that's the difference between the mono and lophotrichous. Then another query is how can organisms be classified based on 16S rRNA sequencing. So this is actually a molecular level classification, or we classify the DNA based on the 16S rRNA sequence. So we know that prokaryotic ribosome has around the size is around 70S, and it is made up of two subunits. The largest subunit is 50S, which again contains two RNAs, 23S and 5S. And then we have the smaller subunit, which is 30S, and it contains the 16S rRNAs. And then this particular 16S rRNA gene is widely used as a genetic marker for studying bacterial phylogeny and taxonomy due to several factors. Number one, it is present in nearly all bacteria and has an adequate length of around 1500 base pairs. And this length makes it well suited for bioinformatics analysis. This particular region gene contains very highly conserved regions which have changed very little over evolutionary time. These sequences are used for designing universal primers, and it contains variable regions as well, whose sequence varies from bacteria to bacteria. So in classification, we also need the variation to be present. But again, a gene which becomes variable more often, which becomes very easily mutated, is also not good as a classifier. So these genes offers us both the conservation and balance between the conservation and variation. So very closely related bacteria will show lesser degree of variation in this region, while more distantly related bacteria will show variation in this region. And this variation is very important in establishing bacterial phylogeny. So in between, let me check once more if any queries here in YouTube. So there is no any query in the YouTube, and I hope I am audible. So Queen, you can confirm whether my voice is reaching there or not. Okay. Is it audible now? Okay,
yeah,
some.
it is audible, sir.
Okay, thank you. So let me now try to look into some of the other queries. A very basic question. Another person is asking what is bacteriology. So the word logy in any subject comes from the word logos, which means study, and bacteriology is the branch of science that studies bacteria, and this deals with the studies of the bacterial morphology, bacterial ecology, genetics, and biochemistry, as well as many other aspects such as the identification, classification, and characterization. It's a very important subject, and again, it can be divided into so many specific branches. For example, we may have, medical bacteriology, particularly which deals with disease of humans, disease-causing bacteria of human. Then we may also have plant bacteriology, which are associated with the plant kingdom. And of course, not all bacteria are harmful to us. Many of the bacteria are also beneficial to us. So, in many aspects, we will find out the instances, we will find out the relationship of bacteria with various organisms. So, in certain cases, we study relationships which are mutually beneficial. For example, relationship. So there is a disturbance. Okay. Anyway, so let's try to take up another question which was asked by another participant. The question is why cyanobacteria often called blue-green algae, even though they are prokaryotes. So the term blue-green algae is used in common usage, and it's actually a kind of an informal usage. This is basically common name for all cyanobacteria, even though these organisms are prokaryotes. And it is basically due to a historical kind of an usage, and actually a misnomer, which arises due to the physical resemblance of cyanobacteria to eukaryotic algae. Like algae, cyanobacteria perform oxygenic photosynthesis using chlorophyll A, and it produces oxygen as a byproduct. And these species are mostly found in freshwater, marine, and moist terrestrial environments, just like algae. Many of these cyanobacteria form colonies of filaments resembling multicellular algae. So, however, unlike algae, cyanobacteria lack a nucleus and membrane organelles, due to which they are actually put under prokaryotes. So, let me check once more any queries in the YouTube. No queries in YouTube. Okay, 6 are watching now. So, okay, just give it. Anyway, so let me go to the next question. Another question one of the participants is asked is what is quorum sensing? So here we basically are discussing about communication systems which the bacteria use for coordinating their activities. So these rely on small molecules that help in the signaling for interaction-specific proteins to activate gene expression when the bacterial population reaches a certain level. So there are main 3 types of quorum sensing. One is the Lux R type, present in the gram-negative bacteria, which uses acyl-phosphorylate electrons or AHLs as signal molecules. So kindly refer to the lecture where we have more detailed discussion of this particular Lux type of quorum sensing. Then there is a Another type of quorum sensing, which is oligopeptide 2-component system, present only in gram-positive bacteria, and these uses small peptides as signals. Then we have a third type, the LUX-S AI2 system. These are present in both gram-negative and gram-positive bacteria, and these uses a different type of autoinducer molecule called AI2. So, in another question related to these about quorum sensing, these LUXI-LUXR systems controls these quorum sensing. These particular protein called LUXI produces AHS molecules which will move in and out of the bacterial cells. When the bacterial population is low, the AHS concentration is too low to trigger gene activation. As the bacteria multiply, the ASL level will increase, and they will reach a critical threshold after some time, and they bind to the LUXR protein. This activates LUXR AHL complex, and then it switches on specific genes. So this leads to the coordinated bacterial behaviors like biofilm formation, biovalence, or the production of bioluminescence. Each bacterial species produces its own ASLs, and these therefore ensure that only members of the same species respond to their specific signals. So in a way, a community of bacteria speaks in their own language, and therefore there is no any leakage of information to other bacterial communities. So this is something very, very interesting. So let me check once more about some queries coming out in YouTube. There are no any queries on YouTube. Okay. Right. So another query was on the epigenetic gene regulation. Specifically, the participant has asked us how does epigenetic regulation works. So, as the word epigenetic stands for beyond genetics. So, this is something which is beyond the genes. So, we know that our genome has only 1% information which produces proteins, and remaining are junk. But then now we have found out that large quantity of junk DNA has also very important biological roles. So this epigenetic regulation refers to the control of the gene expression without altering the DNA sequences itself. It involves chemical modifications to the DNA, histones, and RNA molecules. These regulation results from environmental factors, and these are also part of the novel development. So we can regulate by altering the accessibility of a gene, which can be brought about by any of the following methods, like DNA methylation, which leads to gene silencing by preventing the transcription factor binding or repressive proteins. Then we will go for histone modification, which leads to changes in chromatin peptiding and changing gene accessibility. Then comes the RNA interference, which involves the binding of non-coding RNAs to proteins, DNA and RNA, which brings about gene silencing. So in another question, which was asked, how can we determine protein sequence? So these are very established techniques whereby we can determine the order of amino acids in a polypeptide. And for these we have at our disposal several biochemical and analytical techniques. And some of the widely used approaches are number one, admin degradation, and then we have also mass spectrometry. So any standard biochemistry book, you'll find about the advanced degradation methods. Basically, this removes and identifies the internal amino acids from a peptide using phenyl isocyanate and applying stepwise protein sequencing. The amino acid is cleaved at every step, and then this is analyzed by chromatographic methods or by spectroscopic methods, and we can determine up to 50 residues at a go. And then we use modern high throughput methods like mass spectrophotometry, which is mostly widely popular technique, where we use protease dilation. Then MS analysis, multi-top, time of flight, for peptide fragmentation, and bioinformatics comparison against protein databases to determine protein sequences. So these are some of the queries, and these has been already addressed to the particular participants. So let me try to find out any other queries coming live. There is no any queries coming as of now. So yes, so I think we can wait for some time if some people would like to join and ask something. Okay, you can tell us how to go ahead from here. Chris has something. Chris wanted to say something. Yeah, you can unmute and ask me, please.
I've got an out-of-class question. In Australia, they would put glyphosate, Roundup. It's a poison that's really it's a herbicide, and it affects the shikimate pathway in eukaryotic cells and prokaryotic cells. And I'd like to know if you have any information on that, please, because in Australia, they like to squash a lot of things, and it's very favourable here, and I'm into organics and promoting micro growth, not into hindering. So that was my little question. Do you have anything on glyphosate related to microbes? Any papers or anything like that? I know it would be hard off the top of your head, but that's my question. Thank you.
Okay, I think as I can understand, you're speaking about some kind of microbes or toxin which affects shikimate pathway.
Yeah.
Right. Yeah. So no, I don't have, and this is some problem you see there in particular ecological niche in Australia. You are telling, or is that?
It's the government love to use glyphosate. They promoted it, so all the farmers use it. Everyone uses it, and I'm just. It's terrible.
Glyphosate, that is Roundup.
Yes.
Glyphosate, pesticide.
Yes.
Yeah. I think it's a very specific question. I really don't have much details, but as I know, that's a weedy side. If I'm correct.
Correct. Yeah, herbicide. Yeah.
Yes. Glyphosate. Yeah. And then actually, it affects a lot of the soil microbiota, and probably some studies on the endophytic composition of plants also has been conducted by some researchers. Because when you spray it, it also it actually goes to the groundwater as well. So a lot of health concern also there. Yeah. But yeah, I mean, if you have any particular query on that, you could type it into the YouTube thing, or you can also email me. I can definitely, make a study and get back to you with more details.
That would be excellent. Yes.
Yes.
In the half-life of it, the people's brain, they don't even know any information on it. And it's, I know more about it than them, and it's really unfortunate. It shouldn't be like that. But they should be professional, but it's just the way that Australia does it. They don't, the government uses it a lot here, so you know, they've got to be careful with what is spread. So yeah, but I'm a scientist, so I understand pathways and how things actually work, and it's not looking good for glyphosate. But yeah, so I'd like to contact, so yeah, if we could, I've written something in the meeting chat here, I don't know if that helps. So
yes, so we have some modules already recorded in the future about these impact of all these pesticides in environment. And of course, the microbes also has evolved, and some of them have gathered the capacity to decompose many of these pesticides. And weedicide is one of those kind of pesticides, because we consider weeds as pests, they're plant pests affecting our plantation crops or our cultivated crops. So, and in fact, there are approaches where people have been trying to build up pesticide resistance in the target crop, which we want to grow commercially, so that they are not affected when the pesticide is applied. Weedicide is applied. And but of course, we know that any chemical is a potential hazard to the microbial world. But there are some pesticide microbes which can degrade many of these pesticides, and that's an interesting branch of study. Right. So please write the specific query to me, and I will try to. This is exactly not part of this basic module. Yeah, only we're trying to give the concepts, the taxonomic concepts. You know how the genes are expressed. And you know how we control the growth of microbes by physical, chemical, biological methods. So trying to lay the foundation for the future course of discussion. Yeah. I hope this satisfied some of your queries.
Yes, it's good, thanks. I actually also co-chaired Cyanobacteria. I've been doing that for quite a few years. That's an interesting discussion for plant science. Arthrospiricotensis.
Okay. So there are no any other queries in YouTube channel, as I can see. So Queen, if as there are no other queries, I think we can stop here, or we can also wait, whatever you suggest.
Yes, sir. If you want to wind up, you can wind up.
Okay, then many people raised certain queries, and I think they are not present in this interaction. Anyway, I think this is a recorded session, so when they log back or log in some other point of time, you can find your queries. We did not take your names specifically, but those have also been posted to you. Kindly go through the material that has been sent to you. And with this, I come to the end of this interactive session. Thank you.